The histology of gastrointestinal tract. This is going to be a series of videos explaining the histology of various parts of the GIT. In this video, we will mainly focus on the general structure of the gastrointestinal tract. The portion of the alimentary canal that extends from the proximal part of the esophagus to the distal part of the anal canal is a hollow tube of varying diameter. This tube has the basic structural organization throughout its length. Its wall is formed by four distinctive layers. You can see that in the slide. From the lumen outward, they are first one mucosa, submucosa, muscularis externa, and serosa. The mucosa, consisting of a lining epithelium, an underlying connective tissue called lamina propria, and the muscularis mucosa composed of smooth muscle. The submucosa consists of dense irregular connective tissue. The muscularis externa consists in most parts of two layers of smooth muscle. The serosa or adventitia. The serosa is a serous membrane consisting of a simple squamous epithelium, the mesothelium, and a small amount of underlying connective tissue. The adventitia consists only of connective tissue, which is found where the wall of the tube is directly attached or fixed to the adjoining structures, that is the body wall and certain retroperitoneal organs. Now we will discuss some points regarding this particular diagram. This diagram shows the wall structure of the alimentary canal in four representative organs: the esophagus, stomach, small intestine, and large intestine. The villi, that is a characteristic feature of the small intestine, they are not present in other parts of the alimentary canal. Mucosal glands are present throughout the length of the alimentary canal, but sparingly in the esophagus and oral cavity. Submucosal glands are present in the esophagus and duodenum. The extramural glands, that is the liver and pancreas, empty into the duodenum, that is the first part of small intestine. Diffuse lymphatic tissues and nodules are found in the lamina propria throughout the entire length of the alimentary canal. It is shown here only in the large intestine. The nerves, blood vessels, and lymphatic vessels reach the alimentary canal via the mesenteries or via adjacent connective tissue, that is the tunica adventitia, as in the retroperitoneal organs. First, we will discuss about the mucosa. The structure of the esophagus and gastrointestinal tract varies considerably from region to region. Most of the variation occurs within the mucosa. The epithelium differs throughout the alimentary canal and is adapted to the specific function of each part of the tube. The mucosa has three principal functions: protection, absorption, and secretion. The histologic characteristics of these layers and their functions are described in the following slides in relation to specific regions of the digestive tube. The first point: the epithelial barrier separates the external luminal environment of the tube from the tissues and organs of the body. The barrier aids in protection of the individual from the entry of antigens, pathogens, and other noxious substances. In the esophagus, a stratified squamous epithelium provides protection from physical abrasion by ingested food. In the gastrointestinal portion of the alimentary tract, tight junctions between the simple columnar epithelial cells of the mucosa serve as a selectively permeable barrier. Most epithelial cells transport products of digestion and other essential substances such as water through the cell and into the extracellular space beneath the tight junctions. The absorption of digested nutrients, water, and electrolytes is possible because of projections of the mucosa and submucosa into the lumen of the digestive tract. These surface projections greatly increase the surface area available for absorption and vary in size and orientation. They consist of many Structural specializations. Plicae circularis, villi, and microvilli. Plicae circularis are circumferentially oriented submucosal folds present along most of the length of the small intestine. Villi are mucosal projections that cover the entire surface of the small intestine, the principal site of absorption of the products of digestion. Microvilli are tightly packed microscopic projections of the apical surface of intestinal absorptive cells. They further increase the surface available for absorption. The secretory function, it is carried out largely by glands distributed throughout the length of the digestive tube. The various secretory products provide mucus for protective lubrication as well as buffering of the tract lining and substances that assist in digestion, including enzymes, hydrochloric acid, peptic hormones, and water. The mucosal epithelium also secretes antibodies that it receives from the underlying connective tissue. The glands of the alimentary tract develop from invaginations of the luminal epithelium, and they include mucosal glands, submucosal glands, and extramural glands. The mucosal glands will extend into the lamina propria. Submucosal glands either deliver their secretions directly to the lumen of the mucosal gland or via ducts that pass through the mucosa to the luminal surface. Extramural glands that lie outside the digestive tract and deliver their secretions via ducts that pass through the wall of the intestine to enter into the lumen. The liver and the pancreas are extramural digestive glands that greatly increase the secretory capacity of the digestive system. They deliver their secretions into the duodenum, the first part of small intestine. As previously mentioned, the mucosal glands extend into the lamina propria throughout the length of the alimentary canal. In addition, in several parts of the alimentary canal, that is in the esophagus and anal canal, the lamina propria contains aggregations of mucus secreting glands. In general, they lubricate the epithelial surface to protect the mucosa from mechanical and chemical injury. These glands are described in the following slide in relation to the specific regions of the digestive tube. In segments of the digestive tract in which absorption occurs, principally the small and large intestines, the absorbed products of digestion diffuse into the blood and lymphatic vessels of the lamina propria for distribution. Typically, the blood capillaries are of the fenestrated type and collect most of the absorbed materials or metabolites. In the small intestine, lymphatic capillaries are numerous and receive some absorbed lipids and proteins. The lymphatic tissues in the lamina propria function as an integrated immunologic barrier that protects against pathogens and other antigenic substances that could potentially enter through the mucosa from the lumen of the alimentary canal. The lymphatic tissue is represented by the diffuse lymphatic tissue, lymphatic nodules, eosinophils, and other cells, macrophages and sometimes neutrophils. The diffuse lymphatic tissue consists of numerous lymphocytes and plasma cells located in the lamina propria and lymphocytes transiently residing in the intercellular spaces of the epithelium. Lymphatic nodules are well-developed germinal centers. They have well-developed germinal centers. The diffuse lymphatic tissue and lymphatic nodules are referred to as gut-associated lymphatic tissue or GALT. In the distal small intestine, the ileum, extensive aggregates of nodules called Peyer's patches occupy much of the lamina propria and submucosa. They tend to be located on the side of the tube opposite the attachment of the mesentery. Aggregated lymphatic nodules are also present in the appendix. The muscularis mucosa, the deepest portion of the mucosa, it consists of smooth muscle cells arranged in an inner circular and outer longitudinal layer. Contraction of this muscle produces movement of the mucosa forming ridges and valleys that facilitate absorption and secretion. This localized movement of the mucosa is independent of the peristaltic movement of the entire wall of the digestive tract. Now we can move on to the details of submucosa. The submucosa contains the larger blood vessels that send branches to the mucosa, muscularis externa and serosa. The submucosa also contain lymphatic vessels and a nerve plexus. The extensive nerve network in the submucosa contains visceral sensory fibers mainly of sympathetic origin, parasympathetic ganglia, and preganglionic and postganglionic parasympathetic nerve fibers. The nerve cell bodies of parasympathetic ganglia and their postganglionic nerve fibers represent the enteric nervous system, which is the third division of the autonomic nervous system. This system is primarily responsible for innervating the smooth muscle layers of the alimentary canal and can function totally independently of the central nervous system. In the submucosa, the network of unmyelinated nerve fibers and ganglion cells, they constitute the submucosal plexus, which is also called as Meissner's plexus. As noted, glands occur occasionally in the submucosa in certain locations. For example, they are present in the esophagus and the initial portion of the duodenum. In histological sections, the presence of these glands often aids in identifying the specific segment or region of the tract. The muscularis externa. In most parts of the digestive tract, the muscularis externa consists of two concentric and relatively thick layers of smooth muscle. The cells in the inner layer form a tight spiral described as a circularly oriented layer. Those in the outer layer form a loose spiral described as a longitudinally oriented layer. Located between the two muscle layers is a thin connective tissue layer. Within this connective tissue lies the myenteric plexus, also called Orbach's plexus. It contains nerve cell bodies, ganglion cells, of postganglionic parasympathetic neurons and neurons of the enteric nervous system, as well as blood vessels and lymphatic vessels. Contraction of the inner circular layer of the muscularis externa compresses and mixes the contents by constricting the lumen. Contraction of the outer longitudinal layer propels the contents by shortening the tube. The slow rhythmic contraction of these muscle layers under the control of the enteric nervous system produces the waves of contraction, which is known as peristalsis. Peristalsis is marked by constriction and shortening of the tube, which moves the contents through the intestinal tract. A few sites along the digestive tube exhibit variations in the muscularis externa. For example, in the wall of the proximal portion of the esophagus, that is the pharyngoesophageal sphincter, and around the anal canal, that is external anal sphincter, striated muscle forms part of the muscularis externa. In the stomach, a third obliquely oriented layer of smooth muscle is present deep into the circular layer. And in the large intestine, part of the longitudinal muscle layer is thickened to form three distinct equally spaced longitudinal bands called tinea coli. During contraction, the tinea facilitate shortening of the tube to move its contents. At several points along the digestive tract, circular muscle layer is thickened to form sphincters or valves. And from the oropharynx distally, we can see mainly four such structures. The pharyngoesophageal sphincter. Actually, the lowest part of the cricopharyngeus muscle is physiologically referred to as the superior or upper esophageal sphincter. That is the pharyngoesophageal sphincter. It prevents the entry of air into the esophagus. The next one is inferior or lower esophageal sphincter. As the name implies, this sphincter is located at the lower end of the esophagus. Its action is reinforced by the diaphragm that surrounds this part of the esophagus as it passes into the abdominal cavity. It creates a pressure difference between the esophagus and stomach that prevents reflux of gastric contents into the esophagus. Abnormal relaxation of the sphincter allows acidic content of the stomach to return, that is reflux, into the esophagus. If not treated, this condition may progress into GERD, which is also known as gastroesophageal reflux disease. It is characterized by inflammation of the esophageal mucosa, strictures, and difficulty in swallowing, that is dysphagia, with accompanying chest pain. The next one is pyloric sphincter. Located at the junction of the pylorus of the stomach and duodenum, that is the gastroduodenal sphincter, it controls the release of chyme, the partially digested contents of the stomach, into the duodenum. Next one, ileocecal valve. Located at the junction of the small and large intestines, it prevents reflux of the contents of the colon with its high bacterial count into the distal ileum, which is normally has a low bacterial count. Last one, internal anal sphincter. This, which is the most distally located sphincter, surrounds the anal canal and prevents passage of the feces into the anal canal from the undistended rectum. Finally, the serosa and adventitia. The serosa is a serous membrane consisting of a layer of simple squamous epithelium called the mesothelium, and it also includes a small amount of underlying connective tissue. It is equivalent to the visceral peritoneum described in gross anatomy. The serosa is the most superficial layer of those parts of the digestive tract that are suspended in the peritoneal cavity. As such, the serosa is continuous with both the mesentery and the lining of abdominal cavity. Large blood and lymphatic vessels and nerve trunks travel through the serosa from and to the mesentery. They travel to the serosa to reach the wall of the digestive tract. Large amounts of adipose tissue can develop in the connective tissue of serosa. Parts of digestive tract do not possess a serosa. These include the thoracic part of the esophagus and portions of structures in the abdominal and pelvic cavities that are fixed to the cavity wall, the duodenum, ascending and descending colon, rectum, and anal canal. These structures are attached to the abdominal and pelvic wall by connective tissue, which is known as the adventitia, and it blends with the connective tissue of the wall. With that, we are completing the general histological or microscopic structure of the gastrointestinal tract. In the coming videos, we will discuss individual region one by one. Thank you for your patient listening.